Thanks for coming to the Thursday seminar series. Before I start, I wanted to share with you all that the college has recommended tenure and promotion to our dear friend Natalia Umania. And Gideon Bradbird. Gideon's not here. He had to rush home for childcare, but he's sorry to miss your talk, he told me. Many folks in this room know Nick Gotelli as one of the great community ecologists on the planet. I'm sorry? Feedback. There's feedback? Now you're better. Many people know Nick Gotelli as one of the great community ecologists. Many people know AMA as my most fierce critic. You might know Nick because of his book, Primer of Ecology, that grad students in the grad ecology class all take. You might know him as a co-author with Gary Graves of the book Null Models in Ecology, which I think is one of the classic books in all of ecology. You might know him because of his incredible research that he'll tell you about in just a minute. You might know him for his book on quantitative approaches to doing ecology. You might know him as the person who didn't accept you as a postdoc when you applied. Oh wait, that's only me who knows him as that. Actually, I thought that other people had experienced that, but that's only me. AMA and I are lucky enough to rank Nick among our best colleagues and collaborators ever, and a dear, dear friend. So it's a treat to have him here. I hope you all got to interact with him and get to interact with him. And I'll turn the mic over to Nick Gotelli. Thank you very much.
Great. Thanks so much. I'm so glad to be here. Check my CV. The last time I was here for a seminar was in 1990. So thank you for having me back. So I'm pleased to be able to speak to you today. The title of my talk is the Ecological Impacts.
Yeah, maybe further down.
Yep.
No. See, I know.
Okay. Better? I think so. All right. The ecological impacts of nitrogen deposition, insights from the carnivorous pitcher plant, Sarracenia purpurea. I'm going to start where we usually end these talks, and that is my acknowledgments. In particular, Aaron Ellison has been my long-term collaborator and friend. So today I'm pleased to talk to you about really 30 years worth of research on Sarracenia. I also want to acknowledge the important work of my graduate students, especially Amanda Northrup, Amy Wakefield, and Lindsay Pett. You'll hear pieces of their work in this talk. And also Brian Balliff, a faculty member in my department who works on proteomics, and that will come into the story a little bit near the end. I'll start with this quote from Flannery O'Connor: "The longer you look at one object, the more of the world you see in it." And that will be the theme for this presentation. I'm going to tell you about this remarkable single organism. We're going to look at it at a couple of different levels, and that's the pitcher plant Sarracenia. The unifying theme about this are the varied effects of nitrogen deposition in this system. And we're going to examine several different levels of biological organization to understand the consequences of nitrogen deposition. So here's an outline. I'll start briefly with the nitrogen itself, tell you some things about the natural history of Sarracenia, and then we'll begin starting at the level of stoichiometry and growth responses of individuals with respect to the nitrogen. We'll scale that up to look at the population dynamics and extinction risk, and end up talking about this system as a model for understanding aquatic eutrophication and ecosystem collapse. And I'll close with one single slide about future work of something I'm really excited that I hope we can get funding to do. So let's start with the anthropogenic nitrogen deposition. So lots of interest in the accelerating rate of environmental change. There are ultimate and proximate drivers of that change. This graph is the ultimate driver. This is the graph of the trajectory of the human population. I like this slide because it's so old. 2024 was a projection of a human population size of about 8 billion, and we're right on track with that, heading towards a maximum somewhere between 10 and 12 billion over the next 50 years or so. The proximate drivers from that are quite complicated, but the one I'm going to talk about today is the idea of the burning of fossil fuels, which has released nitrogen oxides, and the use of synthetic fertilizers, releasing ammonium and nitrate. This anthropogenic nitrogen deposition has increased to levels that are 1.5 and 2 times compared to the background ambient levels. And this form of nitrogen is biologically reactive. Plants and also microbes can take it up immediately. Most of the current emphasis on environmental change has been on climate change, but this increase in nitrogen may have even more profound effects on plant and animal communities. The problem is a global one with a great deal of spatial variation in the intensity of this deposition. And in some parts of the world, like Vermont and also where we are right now, that deposition is quite high. Let's look at this organism, Sarracenia, the northern pitcher plant. Carnivorous plants as a group are well known, but surprisingly poorly studied. Carnivory in plants is phylogenetically diverse. It's evolved many times. And these organisms, these plants have several adaptations for attracting, capturing, and digesting arthropods. Not too surprisingly, they are common in low nitrogen habitats because they have switched to getting nitrogen in this different way. They tend to be poor competitors for light and nutrients. Sarracenia itself is a perennial plant, at least in the northeast, of low nitrogen peatlands. For its small size, it's quite amazing that its lifespan is 30 to 50 years. So in many ways, this is like working on a population of trees. They do capture arthropod prey in these water-filled pitchers. It's kind of a low-grade trap, but it does the job, and that is the way they get their sources of nitrogen. But there's a second group of animals associated with this, a diverse community of inquilines that actually uses this pitcher as a habitat to live in. The plant itself has some adaptations for capturing prey and digestion. The red color of the leaves, at least in full sun, is thought to attract insects. There's a primitive nectar that's kind of secreted near the top of the hood. And on the right, you can see there are a series of recurved hairs pointing down on the inside of the tube that make it difficult for insects to crawl back out when they are trapped. Looks like a good trap. It's really not very efficient. Only about 1% of encounters with insects result in a successful trapping. But the insect will be trapped and drown and die in this pitcher. Many tropical pitcher plants have digestive enzymes to break down this prey and release nutrients. Sarracenia does not. Instead, though, what it has is this associated group of inquilines. These are also arthropods, but they live one particular phase of their life history inside this specialized habitat. And some, like the mosquito larvae, this is the only habitat that they occur in. And so what we have contained in a single pitcher plant leaf is an entire miniature ecosystem. This is a brown food web that has captured insect prey at its base. Many of its other food web properties are typical for larger webs that you may be used to thinking about. And it served as a terrific model system for studying interactions at the whole ecosystem level. I'm going to focus more on the plant in this lecture. We will talk near the end a little bit about some of those ecosystem level interactions. The plant, in addition to making that pitcher tube out of its leaves, can also, under certain circumstances, make a leaf called a phyllodes. These are flat. They don't obviously capture prey because there's no structure there to fill with water. They have a higher concentration of chlorophyll and stomates, and they are photosynthetically more efficient than the pitchers are. The plant puts up this beautiful flowering stalk, a single stalk per rosette, and it will only flower after about 3 to 5 years. Remember, it's going to live to 30 to 50 years. It is bumblebee and fly pollinated, and there's relatively short distance dispersal of the seeds. They really don't travel very far away from this flowering stalk. Okay, the leaves persist for about 2 or 3 years, and then they'll start to break down. There's a new whorl of leaves that's produced each spring, and annually the rosette will increase in diameter. Why is this a good organism for studying nutrient uptake? Well, One thing is we don't have the big below ground problem that we face with most plants. This plant has very limited root growth. It's growing in anoxic waterlogged soils. There's basically no uptake of water or nutrients from below ground, and there's very little below ground growth. And that greatly simplifies our job because we can look above ground. This cup-shaped leaf fills with rainwater, and it captures prey that way. That's one source of nitrogen. But it's also picking up precipitation. Snow and rainfall and whatever nutrients are contained in them are also entering the plant through this same pathway. And so because of those features of its biology, it makes it an ideal biosensor of atmospheric nutrient deposition. And I'll show you several ways that we'll be able to see the consequences of nutrient inputs into this system through the cup-shaped leaves. All right. Let's start first with the idea of stoichiometry and growth responses to nutrients. And I'm drawing here in part on the work of my graduate student, Lindsay Pett. The key thing I want to show you in this next part of the talk is that these nitrogen additions alter the growth and the morphology of the plant in pretty dramatic ways. And the way we do this is we take out the existing liquid and we replace it with a nutrient cocktail in which we specify how much nitrogen and phosphorus this plant is going to receive. And then we wait and we measure some attributes of the plant, including the size of the pitcher opening, the keel or flat part of the leaf. And we can turn those into measures of the relative keel size here on the x-axis and the relative tube diameter on the y. So on this axis here, I've arrayed the different treatments that we have set up. And what you'll see is that as we add more nitrogen and more nutrients to the system, we shift down this slope, moving from a round tube that fills up with water towards a flat phylloed leaf. So it's a transformation of the leaf from this trapping structure to this flat phylloed. It's not an on-off switch, but the more nutrients we put in, the more we shift over to this format. And if you want to, you can think of this plant as a highly intelligent optimal forager. Because when nutrient levels are low, it forms the cup, so it's harvesting nitrogen. As nitrogen levels increase, it shifts over to fixing carbon through more photosynthesis. A good strategy in a variable environment. Okay, so there are other knock-on effects that we see from that. The plants do produce more of these phyllodes as we add nitrogen. So we're seeing this phenotypic shift from carnivory to more photosynthesis. And there's also an increased probability of flowering, at least in the short run. I want to contrast that now with the effects of this anthropogenic nitrogen, this atmospheric source, versus nitrogen that comes from the prey that these organisms consume. And I'm drawing here on the master's thesis work of my student Amy Wakefield. And so what we did here was to set up an ecological press experiment. We supplement the ambient or background prey by feeding houseflies that have been frozen and measured out. And we apply treatments, 2, 4, 6, 8, 10, 12, or 14 houseflies every week stuffed into a single pitcher plant leaf. And at the end of the season, we harvest the plants and take our measurements, similar to what we did before. I worried a lot about this experiment when we set it up. What I worried was we were going to completely overload the system and that all of the important activity might really be happening down in this area. And we just didn't know till we ran the experiments. We definitely overloaded the system in the field. These treatments over here, you can smell these from about 10 or 15 feet away, okay? But to my surprise, the plant did not shut down its activity. Instead, a couple things happened. First, we did not see this alteration in the growth form. We did not produce a phyllode as in the previous experiments, even though we're adding plenty of nutrients, this time in the form of prey. But what does happen with all that added prey is the plants are taking up more and more nitrogen. And it's really impressive to me that the system didn't shut down. And the more prey we added, the more nitrogen they took up. We're getting close here to almost 2% nitrogen content in the leaves. That's the point at which things start to maybe become attractive to insect and vertebrate herbivores. At the same time nitrogen is being taken up, phosphorus is also being taken up at these higher levels. You may not be able to see it in this graph, but the phosphorus uptake is a little bit steeper of a slope. And the net result of that is that the N to P ratio drops from ambient as we add prey. And so this is actually suggesting that the plants are now phosphorus limited, not nitrogen limited. Other studies by Terry Chapin previously have suggested that perhaps as much as 40 to 60% of the nitrogen budget of these plants is actually coming from the atmospheric inputs. So it makes sense that now phosphorus is becoming the limiting resource, and we're seeing the differential uptake of it when we offer it in the form of prey. So we have these two contrasting pathways. Nitrogen is important, but it makes a difference whether it's coming from prey or whether it's coming from atmospheric pollution and these other sources. And so what seems to be happening is when we feed them prey, we get the slow release of nitrogen, phosphorus, and also other nutrients that may be present useful for the plant. The anthropogenic nitrogen, that's like the fast food McDonald's diet because it is very rich and immediately can be taken up by the plant. And we are seeing contrasting effects of both of these. It's pretty interesting because over millions of years, Sarracenia has evolved adaptations for low nitrogen environments, but suddenly, in really less than 2 generations, it now may be facing phosphorus limitation, not nitrogen limitation. All right, is this a general pattern? Well, one way we can compare is to look at other plants. So I'm pulling data here from the TRI database. The x-axis is nitrogen content, the y-axis is phosphorus, and I've got a 50% and 95% circle here. So the gray points there are what's in the TRI database of different species of plants and their nutrient profiles. The genus Sarracenia are these triangles. And there's a couple other weird carnivorous plants, mostly Utricularia or bladderworts, that are kind of way up there off in their own space. Sarracenia is within the cluster of what we see in the TRI database, but its N and P content is relatively low compared to other grasses, sedges, and forbs. It's really much more like the nutrient content of evergreen trees. Again, more growing like a slow-growing tree than anything else. Okay, and on a ternary plot with potassium added, we can also see the placement of these species. Same kind of graph, but now I'm showing the results of a couple of different experiments from ours and other groups where people have added prey into the system. And in these graphs, the base here of the arrow represents the control group, and the head of the arrow is the prey addition group. So we're really looking at how the stoichiometry changes relative to this background space. And you can see we certainly are able to push the stoichiometry, but for the most part, the outcomes of these experiments are kind of within the range of what we see of other plant species. And again, on the ternary plot, most of these manipulations are taking place inside this space. Let's do the same sort of graphs, though, now for the other set of experiments where we are manipulating the anthropogenic nitrogen, not adding prey, but actually adding ammonium and nitrate and phosphorus. And here we get a really different result. These manipulations, both by our group and others, are pushing the profiles way out of the boundaries of what we see for most other plants. Again, the control group at the base, the nutrient addition group here. And big elevations in the nitrogen, of course, but also especially in the phosphorus when it's made available in any form. And again, in the ternary plot, these are coming way out. So, as a very general result, we can say that when we add food, the rules, if you will, of stoichiometry are largely obeyed, and the shifts that we see occur within the range of other plant species. But when we add the anthropogenic nutrients, those rules are broken. Now that's at least temporarily. These are all short-term addition experiments. The plant is obviously going to use or reallocate those nutrients that it has taken up. But it is clearly a fundamental shift in how the stoichiometry is reacting to these different sources of nitrogen. All right, let's scale that up and ask, are there consequences for this at the population level? So Aaron and I have worked at a number of bogs throughout New England. I'm going to tell you about 2 long-term study sites where we have monitored plants for, well, 30 years. This is Molly Bog. It's a typical New England kettle hole bog that was scoured out by glaciers. You've got a floating mat of sphagnum. That's the habitat for Sarracenia in this corner of the world. And you've got a black spruce larch forest surrounding the area. So we have long-term plots where we have monitored plants. We, back in '98, tagged 100 adult and 100 juvenile plants at each site, went back every year and measured them. I think the cohort finally conked out about 2021. That was the last year I had plants to measure. We also did seed plantings to estimate recruitment functions. A single flower head has about 1,000 seeds in it, so we plant those out and see what our yields are. And the point of all this activity and all this measurement is to bring everything back to a very simple population growth model. We organize the data into a stage transition matrix, and the arrows represent transitions that can occur from one year to the next. The census data give us the transitions for these. These are mostly probabilities, except this number up here. That's the average number of seeds out of 1,000 in a single flower head that will typically recruit successfully into the following year. And so with all that, as you know, this is not that different than a Leslie matrix. We're going to get a number out of this, the population growth rate, the intrinsic rate of increase of growth. And so here it is. We get it from this matrix transition model in which the population vector is multiplied by the stage transition, and we get this size out at the next step. So here's that all-important number, little r, for the growth rate of these populations. A property of exponentially growing populations is that they have a constant doubling time, so that's a good intuitive way to think about it. So at these rates, these populations would be doubling in size every 125 to 150 years if the growth continued along that trajectory. By way of comparison, the most current estimates for human population growth at the global scale is about 0.0089, a doubling time currently for the human population of 77 years if it were to stay on its same trajectory. So that just gives you a little bit of comparison between the two. Again, we want to know how nitrogen and phosphorus concentrations affect population growth. So the solution here is another one of these experiments in which we are dosing the plants with different nutrient profiles, juveniles and adults. We keep those nutrient concentrations maintained and bracket the observed field values of deposition, following those through a couple of seasons to allow us to measure the transition rates under these different nutrient profiles. All right, so we talked before about the fact that adding nitrogen increases the production of phyllodes and also increases the probability of flowering. Not too surprising, nitrogen and phosphorus are fertilizers, so we expect these positive effects. But at the same time, nitrogen especially is affecting the survivorship of the juvenile plants. And when we're thinking about population growth, we are actually integrating all of these processes together of growth and survivorship and reproduction. So we can do our matrix calculation again, but this time we're doing the calculation for each of the different treatments. It's basically experimental demography that we're doing here. And what you see is that population growth increases for our two control groups. Low nitrogen addition and the two phosphorus additions give us positive population growth. High nitrogen or an increase in the nitrogen to phosphorus ratio push us into the negative territory. We experiment both with the absolute concentration of nitrogen and phosphorus, but the ratio is also equally important because it's the relative availability of those that can determine what's limiting for plant growth. So these are useful, and they do suggest that additional nitrogen added into the system is going to potentially, in the long run, lead to population decline. But in a way, these models are not very realistic because the nitrogen that's deposited every year fluctuates quite a bit. There's short and long-term changes in nitrogen deposition. So it's a bit of a moving target for the plant. And our solution here was to build a more sophisticated transition model in which each year there's a different transition matrix itself. And the way to think about this is in the simple models, we set up this transition matrix, and we just have a simple number that's a constant in there that we use to project population growth. But for this time-varying model, we replace those numbers with an actual function of the nitrogen concentration. And so this allows us to mimic what the conditions are that the plant would be experiencing one year after another. And doing it that way allows us to set up different scenarios. This is really what we want for conservation or projection forecasts, saying, well, what happens if the nitrogen levels stay the same? What happens if they increase? What happens if they decrease? So I'm just giving you one example here. Look at the no change row. That would be, on average, the nitrogen is staying about where it is for the annual deposition. We've got an estimate of, going out 100 years, about a 50-50 chance of extinction for this population. But even under the worst case scenarios, look at how long it takes for populations to go extinct. This is a long-lived, slow-growing plant, so the demographic changes, even severe declines, are going to play out over a very, very long period of time. So here's a graph showing how sensitive these predictions are to the changes in nitrogen. The thing I want to emphasize is how steep this graph is. So we're at about the 50% mark here for the predictions based on the current nitrogen. Slight decreases in nitrogen deposition rates will greatly improve the survival chances for this population, and slight increases are going to make it much more likely that it will go extinct. And I'll come back to this graph in a little bit. A few model updates, and again, I'm going to gloss over a lot of the computational details here. We have built other models incorporating both the effects of NNP on demography, and we have scaled these results up for the entire North American continent. We spent 30 years in these two sites, and we're trying to take the information from those experiments and look a little bit more broadly. So I want to show you this map. This is a spatial, whoops, there we are, spatial variation in the nitrogen deposition rates. Each of those dots is a location of a bog, and what we've got is this nitrogen layer, and this should be no surprise. This is, Upper Midwest, Ohio River Valley. This is the main plume for atmospheric air pollution. And so you can see the very high deposition rates. As we get over closer to the coastline and into the southeast, the nitrogen deposition levels are much lower. So that's sort of the starting point for our projections. Again, I don't want to go into the details, but I do want to look at how the projected values are, and especially emphasize how well they lay out onto this map. So here's our demographic forecast. This is the long-term probability of persistence. So green and yellow are good colors, and the dark blue and black are bad colors. So that's the projected forecast, which would be a pretty grim pattern of extinction over much of the most dense part of the species range. Much better survival as we get out just a little ways over the coast. I really like this graph because it gave us insight into, we've been working for 30 years in two sites, and what we didn't appreciate at the time is when we set this work up, we didn't realize it, but we were on kind of a demographic cusp right here where our sites were. If we had set up a little further to the west, the projections would have been much grimmer towards extinction, but sites a little further to the east, we would have had a much more optimistic scenario to tell. Okay, so that is pretty useful information. That's a demographic forecasting model that is actually built on experimental data. I'm going to briefly contrast this with the way we usually do these forecasts in ecology these days. And that's the use of species distribution models like MaxEnt and other very complicated procedures that use as their basis just the occurrence records where species are found. Again, I won't go into the details, but we've run those models for this same data set. And what I'm circling here in red are the areas of high extinction. And I want to point out what a huge contrast there is between these two forecasting scenarios and suggest that we may be getting into some trouble by just using the easy route of doing the species occurrence records as opposed to 30 years of field work to be able to build a mechanistic model. It's hard to say which of these are true. I'm putting my money on the graph on the left, though, just because we've got experimental data to back it up. But that's a topic for a whole nother seminar, so we're going to let that go for now. I do want to give you a demographic update. We started this work in 1996, and this model that I showed you here was built around the year 2000. I've been going out working at that site every year since, and so I'm going to report on you the current state of things. I wish I had data for some of these, but let me just tell you what I've seen over the past couple decades. First, the habitat has shrunk. There's been a natural movement in of the tree layers as the larch and the black spruce have recruited inward. So there's less area in the open bog for these plants. The density of the plants has gone down. When we first started this work in the bog, you almost could not walk around out there without stepping on a plant. It's much easier now to get out and move around without any plants underfoot. I would estimate maybe a 50% decrease in plant density. Again, I don't have the numbers. It's not how we set the data up. This is just what I see each year I go out. Greatly reduced flowering. Early in the year, in this work, you'd go out to the bog in May, and the field is carpeted with Sarracenia flowers, far too many to even count. And now when I go out to this same site, I see maybe a dozen flower heads across the whole area that I'm looking at each year. And finally, most mysterious of all, there has been a loss of this morphological plasticity. I told you about these experiments where we added atmospheric nutrient and we got those flat leaves appearing. My graduate student, Lindsay Pett, ended up repeating some of this work for her nutrient stoichiometry studies, and she did not get that same plastic response anymore. We don't know why or what exactly has changed, but over that 20-year period, the plants are no longer responding the way they were back in the 90s. So my unfortunate conclusion is that we are actually heading towards a long-term decline in these populations, which sadly is consistent with what the model is. I think it may have to do with more than just the nitrogen, but that is the forecasted update there. All right, I want to shift one more topic with Sarracenia. I want to talk a little bit about eutrophication and ecosystem collapse, and propose to you that this is actually a model system for studying the problem of aquatic eutrophication. So here's what we can do. We can take this plant into the greenhouse and put an oxygen probe in it. And initially, we get this nice periodic pattern of oxygen because, of course, the plant is photosynthesizing in the day and respiring at night. And then we add prey, and that's the blue dots. And we get this very rapid collapse in the dissolved oxygen levels in that tube. And then we get it's really a state change. And now we sort of have a bit of daily oscillations, but now we're down about below 5%. So this, over a very short time interval, is a perfect mimic of what happens to larger aquatic systems that are artificially enriched. There's a slight difference in the detail. Those are enriched with nutrients, which causes an algal bloom, and the death of that algae leads to a big growth of microbes, and the microbe are using a lot of oxygen when that material is broken down. Here, though, we're getting the same effect in this brown food web simply by loading up the system with additional prey. So it's a great model system for looking at ecosystem collapse. And we imagine that there could be some very big consequences of this kind of change. as we possibly make a shift from aerobic to anaerobic bacteria. It was at this stage that I began collaborating with Brian Balliff, a great molecular biologist who specializes in proteomics. We wanted to use the proteomic output of an entire leaf as a kind of measure of its ecosystem function. And we found those proteins could be mapped back to organisms and a database. And definitely there's a big difference between unmanipulated and we use ground up, we call it wasp chow, to feed these plants to do this. So this is terrific. We were excited about the work. Our goal was to see if we could find proteomic early warning indicators of whether systems might be on the verge of collapse. So we pitched this to NSF. This is what we wanted to do in our next work. And we got the grant, and so we started in. It's awesome. But we ran into a problem that we had not anticipated, but we should have. So let me show you this graph, and it will illustrate the problem. On the x-axis of this graph is time. The y-axis is oxygen concentration. And each panel is a different level of nutrient enrichment. The controls are there up at the top, and the high enrichment is at the bottom. And you can see that as the enrichment goes up, we do come into this collapse, which is what we wanted, and that's great. But I want you to focus on this part where the system is initially collapsing. We thought that by just cutting back the nutrient delivery rate, we would slow this process down, and we might have 2-3 days in which the system is on its way to collapse. And so our plan was to collect profiles, look at the proteomes, see if we could find those early indicators, and then do additional experiments with an intervention once that protein marker showed up. But the eutrophic collapse was so rapid we could not do those real-time proteomic assays to identify the biomarkers. Basically, we could not do the grant that we had said we were going to be able to do. So we decided we would, I guess it's not something you'll read about in any of our papers. And it is true that when we do science, we sometimes encounter unknown or unexpected things. But this is something that we really should have known better about. But we did need to go forward. So we decided to look at the back end of this system, that is the recovery from eutrophic collapse, because we noticed it was taking a long time for the oxygen level to return back up after we stopped the enrichment. And there is a large literature on this. It's under the idea of hysteresis, that is the recovery of systems after they've been pushed by an environmental driver. And there's lots of compelling evidence that communities may exist in alternative states that are relatively stable, and they may pass through a tipping point as they move from one to another. So we thought, all right, let's shift gears and let's see if we can't focus on this piece of it. Those states are maintained usually because of a feedback loop that keeps them in one or the other position. And I'm going to draw here on the work of my PhD student, Amanda Northrup. Okay, so simple experimental procedure. What do we need to do? Well, we would load the pitchers with organic matter, measure that twice a day for a couple weeks, also measure the dissolved oxygen, and then plot the organic matter against the dissolved oxygen and get the trace of what we needed. Awesome. Except for this second step. We need to be able to measure the organic matter concentration twice a day for 15 days. It's coming from ants, so we thought, well, we could just fish them out and reweigh them. That wasn't working, so we used our wasp chow. We even came up with this little, tea bag arrangement where we would steep it in the pitcher plant for a couple days and bring it out. Nothing was working. I couldn't get accurate measurements. I got really frustrated. I finally turned to the molecular biologist. I said, look, is there some molecule we could use to do this? Because we're just not working with the stuff. And he got this little twinkle in his eye, and he said, let me get back to you. And a couple days later, what he came back with was bovine serum albumin. If we load that up with DNA and salts, we can get the nutrient profile to look just the same as the arthropod prey. But this bovine serum albumin is soluble in water, so we can take out just a few microliters and get the measurement that we want. And that was what we needed to be able to do this work. So finally, we could set up these treatments. Here on the x-axis is the environmental driver. That's the BSA concentration. The y-axis is the dissolved oxygen. The red line, that's the loading phase. So we're adding nutrients to the system, and it's starting to crash, and the oxygen level comes down. Once it comes down, we stop the nutrient addition, and the black line is the recovery. And that's the classic hysteresis plot, because even though the BSA levels have now gone down, it takes a while for the oxygen to get back up where it was. It doesn't retrace. So that's pretty cool. We have actually established that a single environmental driver will control the hysteresis dynamics. That's not something we can say for certain in most of the other published studies. But it gets even better. We now up the delivery rate in a different treatment. And now we have no more hysteresis. We just get this return. If you're a restoration biologist, this is what you want to see. You stop the environmental degradation, and the system recovers and comes back in the same way before. We cranked the delivery rate up even further and got a really cool surprise. We generated a reverse hysteresis plot where the initial decline is very steep and the system springs back on the other side. So all of this rich dynamics changed only by altering the enrichment rate itself. Nothing else is different between these treatments. We still hope that we could do the proteomic indicators one day, because there are these really cool phylogenetic differences in the map proteins, but that's going to have to wait for another time. So future work, I'm just going to mention one graph that is so awesome because it's an experiment that I dreamed about for 10 years before we were finally able to do it. We had gone into the field, hit these pitcher plants over a press experiment with broad spectrum and narrow spectrum antibiotics, and it's allowed us to suppress the entire microbiome. So we are very excited to see what, if any, the ecosystem level consequences will be in future experiments, which of course would depend on some future funding from NSF. We'll see. Okay, so to summarize, I've talked to you about the effects of nitrogen deposition at the level of individuals. We've gone to the population level to look at extinction risk. And at the community level, we've talked a bit about using this as a model system for proteomic early warning indicators, but more usefully, eutrophication and hysteresis. I'll stop there. I thank you for listening. I'd be pleased to answer any questions.
I feel like I could ask a million questions. That was an amazing talk. But I think the thing that I found really interesting, or like I would love to hear more, is the loss of plasticity you found between the populations from 96 to, I guess, current day. And my questions are, do you think it is evolution, or do you think, like, I don't know, I know that these are long-lived populations, so I wanted to ask what you thought was going on over there.
So no, I don't. It's possible that it's evolution, but we haven't seen that huge of a population decline. This has been barely, not even one generation's length of time from when those first set of experiments were done. So we would have to ask what else has changed during that time. Well, one thing that's happened is there's been just that much additional nitrogen added into those systems, which are relatively closed. We also have had quite a change in climate over that same period. I don't know how much that's going to interact with that, but that's what I would look for to see that. But yeah, we were pretty interested, and Lindsay's a great field biologist, so I don't think this was anything in her experimental technique. I think this is a real result. But yeah, I thought it was pretty interesting and unexpected.
Yes, you mentioned the reduced debt.
Oh, the batteries, yeah, the batteries over there. There you go.
the reduced density for this the pitcher plant. I'm just thinking, like, other carnivorous plants like Venus flytrap, for example, they have a little bit different capture mechanism. Do you think the same effects would obtain for something like that? Yeah.
That's hard to say. Those are facing different sorts of pressure. I'm going to say that they will, simply because I showed you those data where we looked at the other experiments of food addition and also nutrient addition. I don't think there are Venus flytraps in that, but the carnivorous plants as a whole are doing something fairly consistent. So I guess based on what we've seen already, I would say I would expect similar kinds of responses. Okay.
Thank you so much. Wonderful talk. So I was wondering about the availability of nitrogen in light of your results on how with increasing nutrient level, the oxygen level goes down. Because we know in lakes, as the oxygen is depleted, the availability of the forms of the nitrogen change from nitrates to nitrites and then ammonia, and that might change the bioavailability for different organisms depending on what their preference is. I wonder if there is a preference in Sarracenia, and then how would that interaction between the oxygen availability and the nitrogen forms play out?
That's interesting. We've not explored the different forms of nitrogen themselves beyond what we just set up for these experiments. But I could imagine a second set of experiments that are looking at the molecular form that it comes in to see what happens to it. This response was good enough that it seems like I don't imagine it would be just the shift in nutrients. I really thought we would have collapsed the plants, and if you saw them in the field, they were just absolutely disgusting at the high addition level. So it was pretty amazing to me, frankly, that the plant was able to do anything at all under that sort of stress.
Other questions for Nick? Yep. Hang on, I'll come over.
Great, thanks. So if we move back to the inquilines and think about some of your experiments from the perspective of an inquiline, did you measure or observe any changes in inquiline community under your different types of nitrogen addition, say the environmental versus giving them more prey? Because I imagine that it matters to an inquiline what kind of nutrients they're getting and in what combination.
So in some of our experiments, we actually deliberately excluded the inquilines. So the initial nutrient ones that I showed you, once we set the nutrient cocktail up, we removed the inquilines and we plugged the opening to the leaf with spun glass wool, which is really nasty stuff to work with in the field, but it does prevent adult arthropods from ovipositing in the pitcher. But we've gotten similar results in other experiments where we've done whole plot sprayings, and that does not exclude them. So we're seeing similar things there. There may well be an effect on the inquilines. Our research is pointing that the plants and the microbes may be the heavy hitters in this system. One of the experiments I did not talk about today was we fed labeled nitrogen to fruit flies as a source of prey, and then we fed them to the plants to look at the translocation of nitrogen. We set up two treatments, one with the inquilines present and one without any of those macroinvertebrates. And the nutrient transfer rate to the plant is just as high in either case. So although the just so story is that the plant depends on these inquilines to shred the prey and break them down, the reality is the microbes will take over the job if those inquilines aren't there. Thank you.
Any others? Where? In the very front? Yep.
Thanks. I have a pretty simple question about these things don't produce rings, so how do you infer the age? And then secondly, in regard to the plasticity, could it be that you were sort of looking at juveniles the first time you measured and adults later on, and this could have changed?
Yeah. So the age is hard to get. We don't have anything comparable to tree rings or otoliths that we can look at. And so most of it is done by what we know from plants we have monitored. The recruits are easy to identify during that first year. It's just a single little tendril is kind of buried in the shag carpet of the peat moss. But we're working with things that are larger that we estimate are at least 2 or 3 years old from the size of the whorl. They're going to add about 5 to 7 new leaves every year into this whorl as the outer ones will die. So we've really done this only on the basis of the size. But all the plants in both of these experiments were of sufficient size that they would have been in the same ballpark. And we weren't doing, none of those experiments were done with plants that are actually flowering.
All right. Well, Nick, that was great as usual. Just as good as your talk in 1990 that you gave here, I'm sure. But thank you very much for a wonderful talk.
My pleasure. Thanks for having me.